Thank you, Christina. And we all will join forces so you will succeed, I promise. And now let's move forward to the Human Brain Project, the big dream that actually was realized. Professor Idan Segev, who was one of the first to initiate and share this big dream project with Henry Markham, from the Blue Brain to the Human Brain project, and the success, he will present the project. But as Lois mentioned earlier, I just want to mention that there are 5 Israeli groups participating and contributing to the Human Brain Project, to the wrap-up phase now. The Hebrew University in Jerusalem, presented by Idan Segev, the Weizmann Institute, presented by Professor Yadin Dudai and Professor Rafi Malach and Professor Shimon Ullman and some others. The Haifa University, and also presented by I forgot his name, I think. Do you remember? Avi Karni, right? And the Science Museum in Jerusalem, presented by Maya Levi, that will give a lecture on Friday morning, and you are all invited. Tel Aviv University is represented by Professor Yoav Binyamini, which is here with us. And אתה יכול לסגור את הדלת שם? תודה. And myself, we are part of the medical informatic work package that is developing a platform to share, analyze, host, and combine all hospital data, and actually to find the right and reliable translation to the well-being of us all. Personally, I can tell you that this is the great, or should I say the inspiration moment, meeting with all the groups and the students, and this is a real reason for all this Human Brain Project. Now, I think this is what all this work package is about, and what is the Human Brain Project about. Really to converge and bring all the expertise, like was said, together. And the workshop that we are going to be here, the 3 days, will try to present the broad fields of brain research, and the next day, all the discussion about the future of neuroscience, the future of education in neuroscience, the connection between industry and academia, and all these parts that will build the right platform for the right activities in the future. So I wish us all a fruitful and enlightening workshop, and not less important, the outreach to the public, which will be on Friday morning, which I think this is another very important layer in the Human Brain Project. And I'm happy that David is here with us, that will talk about that. I want to invite Idan, a real visionary, a real pioneer in brain research, who established the computational neuroscience, the first in Israel, that turned into the Lili Safra Center for Brain Research nowadays. He sure knows how to dream and how to make it happen. Thank you.
Thank you very much to all of you for being here, for coming, to Mira for the organization. What I thought I will do in the half hour that I have, which is a little bit less now that we are delayed, is to highlight the way I think about what happens in brain research today worldwide. So I would like to start with discussion of some of the key of the key mega projects so that we shall get a perspective about the Human Brain Project. It's not the only dramatic project that is happening today in the world. There are a few dramatic projects, and I would like you to know about them and make a little distinction between the Human Brain Project and others. And then I will speak a bit about my aspect, which I think is the seed of the Human Brain Project that became much broader than the Blue Brain Project. So I want to say a few words about the Blue Brain Project. And if I will have time, I would like to discuss some very exciting starting of a dissemination of the Human Brain Project into the public. So let me start just showing you a few, I think, of the most dramatic attempts today to do an historical act rather than a more classical, daily based, lab work, something much more in a larger scale. I'm sure you must have heard about Allen Institute, because in the last 10 years they did some wonderful work on the on the genome or the gene expression in brain of mouse, and now they completed the human brain gene expression, just published in Nature. Fantastic work and very useful from the medical point of view and others. Now they dwelled into something new, so they have a new $300 million and a new wonderful building to start a whole new project. And I will say a word about that. The first paper from this project just came a month ago in Nature, the mesoscale connectome. I'll say a word about that. The Human Brain Project I'm going to talk about, the brain, or there are names that are changing. It used to be called the BAM project, the Brain Activity Map project by Obama. President Obama announced that, and then now they are growing very fast. I think partly because of the competition with Europe. After all, Europe and America compete who is bigger, and in that sense it's good for the brain because there are all these projects. So the Brain Project I will mention along my talk is really focusing on developing new technologies to record at the level of single spike, single cell, non-invasively from 100 millions or 100 millions of cells simultaneously. And we should discuss why is it so important for the Americans in this case to focus on recording at the level of spikes, at the level of single spikes, single unit from many, many cells. Japan just decided to focus on the monkey brain, so this is a whole new project in Japan, all influenced in some sense by the Human Brain Project flagship. There is the Connectomics worldwide project to try to reconstruct fully at the EM level, the electron microscope level, a whole animal and so forth. And there are also other attempts all over the world. So we are in a historical moment. I think President Peres realized that by giving a prize, $1 million every year to a brain researcher. There is an excitement, very special excitement at that time, which is lucky for you, the young generation, but also lucky for us, the less younger generation, because we are feeling that something dramatic is about to happen, and that's a very good feeling. The only thing is that something dramatic should happen, and you have to work for that. And so we are working for that very hard. So this is the Allen Institute, the Mindscope Allen Institute in Seattle. The scientific director is Christoph Koch. They are building this building. We are also in Jerusalem building this building, so you see that there are some wonderful buildings. This is by Norman Foster, and so on. In America, in New York, they are also building a new building in New York. You know, in Harlem, what used to be Harlem will become a new campus for the Columbia University. In this campus, there is also an amazing new building by Renzo Piano. You know Renzo Piano from many places. So things are happening in the world, but this will be probably the nicest in the world. And I want just to tell you one thing about what is the focus, and that's very interesting. So this is the agenda, this is the manifesto of the Allen Institute Mindscope project. So I'm not going to read everything, but you can find it, of course, in the internet. I just want to highlight something because I think it's very interesting what they decided. You know, they were wondering whether they should focus on Parkinson, or whether they should focus on Alzheimer, or whether they should focus on vision or motor system, or what should they focus on. So they gathered a lot of experts along 2 years of discussions, and eventually they decided on that, which sounds very, very strange. We want to catalog and characterize the cellular building blocks of the cerebral cortex. You may know that we don't even know today how many cell types are there in the cortex. And the question is, how do you define a cell type? But we are not going to go into it, but it's not clear who are the building blocks of the cortex or any other system. We really don't know. So that's one of the issues, is to catalog and characterize the cellular building blocks of the cerebral cortex, their dynamics, cell type specific structure, function, connectomes. This is a key word that you are not going to escape from in the next 10 years, because it's clear that in the next 10 years, people are going to slice pieces of brain or whole brain. There is new developments in this field of connectomics. And this is one thing that they want to do, is really to understand the roadmap, the full connectivity, in this case, of the visual system of the mouse. We want to know what the animal sees, how it thinks, the mouse, and how it decides. I mean, this is very ambitious, yes? You want to know how the animal decides. What does he think? Is he in love with the other mouse and so forth? What happens in this mouse? So this is serious. This is serious. So this is megalomaniac, and that's exactly the issue of this project. These are megalomaniac projects. They don't want to do what was done before. They want to do something dramatically new. And this is one of them. And it's going very well. You really should look at the paper from last month, where they succeeded to start reconstructing anatomically, not at the microscale level, not at the EM level, but at the mesoscale level, with new technologies to reconstruct long-range connectivity at single axon level and so forth. Very, very dramatic. But eventually everybody wants to do it. You know that maybe you know Janellia Farm. Janellia Farm is focused on now slicing the fly completely. The fly has 850,000 cells in the brain. They want to slice EM-wise the whole, and they develop new technologies to get something that looks like that. So this is from the cortex of a mouse, very small piece, 20 microns on 20 microns on 40 microns. But this is what you should expect when you cut a piece of a brain at the EM level, and you get this connectomic. You see the blood vessels, you will see the synapses, you see dendrites, you see axons, you see everything. Of course, you may ask yourself, so what comes out of this information? We shall discuss it. But this is a huge attempt today to cut pieces of the brain at the EM level and to know who is connected to who, by which type of synapse. Is it inhibitory, is it excitatory, and so forth. Connectomics. It's a huge, huge attempt that you will see more and more. In Israel, we don't have the technology to do that, but in Heidelberg or in Janellia or in Harvard, they have all these new technologies to slice the brain, automatically reconstruct it using computer vision and things of that sort. Very dramatic. Now I want to say a word about the Obama project, just to give you the perspective. So Obama focus is really speaking about the level of spikes, as I said. You know, for example, from the work of HUJI at the Hebrew University, and many of you knows, of course, that when you record from a given region in the brain at the single cell level, you see this spiking activity of cell number 1, of cell number 2, of cell number 3. You see the spiking activity of these cells. This is the signature, as we believe today, of the coding processes in the brain. So the brain codes information, eventually, by spikes. That's the fundamental assumption of neuroscience today, that if I can read the spikes of a brain, and I know the code, I know the language, I can tell you what happens in the brain, completely. What are your thoughts? What are your fears? What do you want to move, or what do you see? I can read the code by looking at the spikes. I don't need more than that. So the level of resolution is believed. Of course, below the spikes, there are genes and there are molecules and there are receptors. But eventually, the assumption of this American project, of this Obama project, is that it's enough to look at the spikes, but many cells together, not only 3 in this case, in order to understand basically everything, to read everything from the brain. You don't need to go below. But sometimes these spikes behave badly. For example, in Parkinson, you see that the same network of cells that are hidden there starts to fire differently. There is something wrong, so to speak, in this activity that manifested as Parkinson. Why this activity suddenly becomes like this? This is not the Obama project. The Obama project wants to be able to monitor the activity of the brain in real life while the animal, I mean us, behave. So we shall have probes in our brain, and these probes will transmit spikes, and you will be able, hopefully, to read these spikes and say, oh, you're about to get epileptic seizure. You're about to get Parkinson. So this is the signature of everything, including diseases, or including love, or including memory processes, and so forth. That's the assumption of neuroscience, and that's what they want to develop. You know that today we don't know how to record spikes. It's spikes without being very invasive. We have to implant an electrode in the brain, as you can see here, and that's something that is unacceptable. And also you don't know how to implant millions of electrodes. But with the nanotechnological world, with optogenetics and other techniques, you may be able to record from many, 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 many millions of cells simultaneously in the real behaving system. So that's the Obama project. We have some proofs of principles. For example, the work of Elon Vaadia is an example of recording from behaving animal while the animal behaves, recording spikes, so implanting electrodes in the motor cortex in this case, recording spikes from this cell or this cell or this cell or this cell. So this is a spiking activity of a single cell. So this is how it sounds when you listen to the spikes from this one individual cell while the monkey is doing some movement. And eventually, the belief is that these spikes that are running here, which usually when this network is coordinated with this particular music of spikes, activate this hand through the spinal cord. If something goes wrong, you may be able to read the spikes directly from the brain and use the spikes to activate a robotic arm. And this is starting to be successful. This is an example of a monkey sitting here with about thousands of electrodes coming from his head, or her head in this case. The hands are tied, so she cannot move the hands, the monkey. But she wants to get food from here. And you can activate the robotic arm directly from the spikes. You record many spikes, and you know the code of the spikes coding from movement. You see that this robotic arm, which is outside of the monkey, is directly activated after some analysis of these spikes. And you can eat, in this case, like we eat when we take our hand, moving our hand from our spikes. So this is why spikes are so important. So this is brain-machine interface. The brain activates a machine. And when I say the brain, I mean the spikes recorded from the brain, is activating this. Now, the problem is that and that I'm coming now to the Human Brain Project. The problem is that, of course, you see spikes in the brain, you suddenly see the spikes goes wrong in Parkinson or suddenly in epileptic seizures. You don't know why. Something goes wrong, something goes out of balance. Is it a synapse? Is it the excitability of a cell? Something goes out of balance. Suddenly there is a new phenomena, let's say bursting cells, and you have Parkinson. But we have no idea why, in a deep sense, because we don't have theory, we don't have modeling. We can say that this transmitter is missing, there is more serotonin, there is less dopamine, there are these things, but how is it related to the code? And so I like this citation by the great physicist William Thomson, who say, I'm never content until I have constructed a mathematical model of what I'm studying. If I succeed in making one, I understand. Otherwise, I do not. I can talk until tomorrow, I can draw graphs until tomorrow, but if I don't have a mathematical model of the phenomena I study, I don't understand. It's a partial understanding. And this is the ultimate proof in neuroscience. That's the most successful theoretical neuroscience ever, is the Hodgkin-Huxley model, because they saw the spike for many, many years. Everybody spoke about the spike, the spike, the spike, but nobody really understood the spike, although there were a lot of attempts to understand the spike, until they wrote these 4 differential equations. After writing these 4 equations, we understand the spike. Why? Because we know the underlying mathematics of this particular phenomena. So I can say that I understand the spikes because I have these equations. I saw the spikes before, so they didn't invent the spike, so to speak, but we did not have a mathematical model of this phenomena. And that's what we want to do in the Blue Brain project. We want to develop a mathematical model of a piece of a brain, understand how this piece of brain generates spikes, how the synapses are responsible for this particular activity or another. Okay. So what do we want to do? And I want to speak about the Blue Brain Project because, as I said, this is a seed and an important part of the Human Brain Project, but certainly not the whole Human Brain Project. You will hear along these 3 years, I'm sure, many, many aspects, and I will mention later if I have time. What do we need to do in order to now build a model, not of a single cell or of a single spike, but a whole network composed of many cells, of many connections, like, for example, the cortical column. So I'm looking at your cortex or the mouse cortex. There is a column there, a structure there, a volume there, let's say of 100,000 cells. And I want to reconstruct mathematically the activity in this column. Certainly I need to know the cell types. As I said before, there are different types of neurons, some pyramidal cells, some spiny-stellate cells, some inhibitory cells, some excitatory cells. We want to know which types compose this particular tissue. But you also want to put these cell types in the correct numbers and the correct location. So you want to put this type of cell, many here, let's say, and this other type of cell there, and this other type cell. You have to place the correct cell types and the correct numbers and densities in particular layers of the cortex. Eventually you build it. Anatomically, we know how to do it, although we don't completely have all the cell types in the cortex, as I mentioned, but we have some of it. Then you have to assign to each cell type a mathematical model describing the spikes that it generates. So you need an extended Hodgkin-Huxley model because the spike, as I will show you in a second, these cells fire in a different type of firing. There are different electrical types, not only morphological types, but also different electrical types. So this type likes to do ta-ta-ta-ta-ta-ta, and this other cell likes to do ta-ta, stop, and so on, but all with spikes, but different spiking pattern. So we need to assign the mathematical model for each cell types related to the property of this cell. So it's a lot of work just to put things together. And that's what we started to do in the Human Brain Project, and a paper was just sent last week after actually 15 years of work. It's a huge paper, it's a mega paper, as everything is megalomaniac in the Human Brain Project. This is also, the paper is megalomaniac because it has 40 figures for nature. This is unusual. Eventually we succeeded in replicating the electrical activity of this piece of tissue with the 6 layers, with all the cell types that we know of. These are the cell types that we know of today existing in the brain, in the cortex of the mouse. These are all the inhibitory cell types, which is about 15% of all types of all cells. There is the excitatory types in the different layers with different proportions and all that. I'm not going to burden you with details. I think you understand the principle that if you really want to replicate mathematically the activity of a piece of tissue, you need to know who are the players, not only morphologically, but also electrically, and then connect them correctly, synaptically. And this is the whole thing of working really on the proportion of each type of the inhibitory type. You know, layer 1 has only inhibition, and layer 2 has a little bit of inhibition and excitation, and the inhibition is built from these types of inhibitory cells, and eventually you get a nice picture, you get a model. So just a word about this is because it's a very important issue that people neglect, especially if they are physicists. Because they assume that a cell is a cell is a cell. So as I can show you here, and these are recordings from the cortex, some cell types, this let's say, when you activate the cell, it tends to fire regularly. Tack, 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 tack. This is the electrical language of this cell. Other cell types, for example, this cell, will fire for the same stimulus, tack, 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 stop. Tack, 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 stop. This is another electrical instrument. It's like in the Philharmonic, when you see it, you listen to this music, not electrical music, but music, then you see some of them are violins, and some of them are trumpets, and some of them are harps. These are different instruments. You cannot assume that the cell is the same thing electrically. And we know that in the cortex there are about 12 types of electrical types. And this also you have to put into building a model of the different types. And this is our work in Jerusalem. We developed a mathematical approach to model different firing patterns of cells. This is the experiment, the brown. the green is the model, you can see that we can replicate not only a single spike with Hodgkin-Huxley equation, but if we extend the Hodgkin-Huxley equation, we can simulate every activity. Tac, 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 stop, tac, 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 stop. It's another differential equation than tac, 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 tac. But we know how to do it, so we know to replicate all cell types today, and that's why we can build a column mathematically. And when you build a column mathematically and you start to simulate it in the mega computer, in the supercomputer that is now in Lugano, you can really now start to mathematically simulate the electrical activity of the whole network, of the 35,000, or we are now at 100,000 of cells, firing with all the axons and all the synapses in the computer mathematically. So that's the mission of the Blue Brain project, is to integrate data, to integrate synaptic data, connectivity data, spiking data, cell type data, to put this together with a simulation tool. And this tool should be, and that's what we want to do, should be now available to the public. If you want to simulate the basal ganglia, if you want to simulate the cerebellum, if you want to simulate the hippocampus, whatever you want to simulate in order to understand signal processing in this tissue, you may use the same platform. You don't need to build a new platform. That's the idea of this, that we shall give you a platform for simulating large-scale tissues, or a single cell if you want. Okay, so I think I explained to you that now I can take this tissue and I can activate this tissue, let's say from the thalamus. I can give it as an input from the thalamus to this cortical tissue in the computer. I activate layer 4, and I'm starting to see some activity. These red spikes are the activity. You can see the activity reverberating in this system, or there are some oscillations, some gamma wave oscillations and so forth developed in this network. Now the question is, when does this network become sick? What do I need to do? What are the regime of parameters that I manipulate in order to make this network Parkinsonian or epileptic, or that the network cannot go to sleep? What are the regime of parameters where you get a phenomena, normal phenomena or disease phenomena? One thing that this building or copying reality, because what we do here is we take biology and we copy it one by one, which is a very elaborate work, but we copy it into a model, and now the model enables me to interact with it, like in the biology, to close this synapse, to activate this cell, and so forth, much easier than in the biology, hoping that I capture the essence of the biology by doing this replication. But I can also analyze in a very modern way what is the intrinsic structure. And you know that there is this modern network theory where you study Facebook networks, friendship networks, disease networks, and so forth, using new network theory. And we can start to use this network theory to ask, for example, is the cortex small world? Are cells connected to each other by, on average, 2 or 3 synapses from this cell to this cell on average, and so forth? We can really start to analyze the network when we have it. So this is the beginning of a quite exciting work, I must say, that I was not aware of it before we started. What amazing things you can do with new theories, like network theories, about networks in the brain. We are also working with the Allen Institute that you heard before. The Allen Institute has working on the mouse brain. And the mouse brain has 75 million cells. We don't yet know the connectivity in this mouse brain, as I told you. We know the mesoscale connectivity. So we know that this region sends axon to this region, and this region sends axon to this region. This is the new paper in Nature that just came from the Allen Institute. But we shall have eventually a connectomics of this particular brain, I guess within a few years, hopefully. And then you can simulate the whole mouse brain. And then you can ask question, what does the mouse see? What does the mouse feel? Because you will simulate the amygdala and you will simulate other regions. So we are approaching this direction. I want just to say a word about human brain, because after all, this is called the Human Brain Project, not the Mouse Brain Project. We are calling ourselves the Human Brain Project, and this Human Brain Project needs human cells. And this is, of course, a rare tissue to get. So there are several groups in the world today that are doing really fantastic work in the anatomy of human brain, including Allen Institute. This is an example from Spain, where he takes dead brains, in this case after accident, people that donated the brain, and you can really reconstruct cells at the level of single spines and dendrites. And you can really start to build anatomically from this the connectivity of the human, let's say, cortex. But we also have work starting in several labs in the world recording from fresh human brain, because these people are taking from in the hospital, they are taking pieces of brain, of human brain, of cortex in this case, because of some tumor that they have to go through, and they take this piece of the brain. It looks something like that. So you see, this is the piece that was taken out from in any operation. It's being done if you need to go down into the brain. And this brain that we have today, let's say this was in Amsterdam. It's not being done in Israel because of ethical issues or religious issues. But this is a piece of a brain that was taken out. This is the size of the brain of a mouse. So you can see that the piece that is being taken out, fresh tissue, is huge. And some labs in the world have started to record, to characterize cell types in the brain of human and so forth. I'm not going to go through it, but this is a cell recorded experimentally, reconstructed anatomically, and we have now a lot of cells reconstructed, recorded, and connectivity already between them. So we are starting to have a model of the human brain, not only of the mouth. I want to complete, because I have 4 more minutes for the half an hour that was given to me. I want to say that there is an issue of too complex model. You sometimes replicate something that becomes too complex to be understood, the model itself. And there are things that needed to be learned from this fellow, Brancusi, who took things and reduced them mathematically, so to speak. So this is the bird, this is the kiss, the minimal kiss that captured the essence of the kissiness. And so that's what we need to do, of course, and there is a whole theoretical part in the Human Brain Project that is trying to reduce the complexity of the models and capturing the essence of the behavior. I'm not going to talk about this. Just to show you that the Human Brain Project is huge. The simulation part that I just mentioned is here. There are many aspects. You will hear about education and you will hear about other things. I just want to say one thing that I was very excited and finish with that, and this was with David. We met in Belgium with a group, and with Maya Levy, who is, you heard about her, she's part of the project, and she's in the museum, the Jerusalem Museum for Science. We realized that one of the role of our project, the Human Brain Project, is to explain to the general public what we do. There is a lot of fear from brain research. You must understand, we are going to read your thoughts, we are going to manipulate your brain, and they are right to be worried. And this is our duty to explain what we do and to discuss the worries. But how do you do it? The idea is to open in each science museum in the world, there are 3,000 science museums in the world, to open a brain corner, a digital brain corner, in each one of these museums. This is our dream. To spread the information from the center about what happens in the world, like I did now in half an hour, to do it systematically, every month or 2 months, send new information to these brain corners all over the museums, and see, interact, enables children and families to come. And you know that the Science Museum are the most visited museums in the world. So in New York, the Natural Museum is much more visited than the Metropolitan, plus the Frick Collection and all others, you know, all together, and the MoMA, all together. This is the place where people are coming with families, and this is the place where we want to teach them about the brain. We want to bring society, or the brain, to the society. We want to demystify the brain and the mind, as if these are two different things, the brain and the mind, as if there is something mystical in the brain. No, it's all physical. We want to humanize brain diseases. And there are other issues that we want to do through this brain corner. So I want to thank you very much for listening. As you see, it's dramatic, it's very demanding, it's very hard to do what we want to do, but if we should sit here in 10 years, either it will be a complete failure, which is possible, but I don't think so. It will be a big success. Thank you very much.